Hello dear viewers, welcome to Mr.Zoologist YouTube channel. Today we are going to discuss about signal transduction. Cell signaling, also known as cell communication, is the process by which cells communicate with each other to coordinate various physiological functions and responses. It means to say cell signaling is essential for all the activities of the cell that are coordinated either from inside or from outside. Cell signaling involves the transmission of signals or messages from one cell to another through a variety of mechanisms. These can be chemical, electrical, and mechanical signals. These signals are typically generated in response to external stimuli or internal changes or internal stimuli, and are crucial for regulating processes such as growth, development, differentiation, metabolism, immune responses, and cell survival. If we talk about the process of cell signaling, ligands and receptors. Signaling begins when a signaling molecule, that signaling molecule is also known as a ligand, binds to a specific receptor protein on the surface of the target cell or within the cell. Ligands can be hormones, neurotransmitters, growth factors, cytokines, or other signaling molecules. And if we talk about signal transduction, upon ligand binding, the receptor undergoes conformational changes leading to the activation of intracellular signaling pathways. These pathways typically involve a series of protein-protein interactions, post-translational modifications such as phosphorylation, and activation of second messengers molecules such as cyclic AMP, calcium ions, or inositol phosphatases. If we talk about the intracellular signaling cascades, activated receptors transmit signals to downstream effector proteins, leading to the activation of various intracellular signaling cascades. These cascades often involve the sequential activation of protein kinases, phosphatases, GTPases, and other signaling molecules, ultimately resulting in changes in gene expression, protein synthesis, enzyme activity, cytoskeletal rearrangements, or other cellular responses. If we talk about the cellular responses, the activation of intracellular signaling cascades leads to specific cellular responses, such as changes in cell proliferation, differentiation, migration, apoptosis, or metabolic activity. These are the responses or feedbacks of the cell that are tightly regulated and coordinated to ensure proper cellular function and adaptation to changing environmental conditions. And if we talk about the types of intercellular signaling, and first of all are the autocrine signaling. In autocrine, auto means self. Autocrine signaling, a cell releases the signaling molecules, autocrine factors that bind to receptors on its own cell surface or neighboring cells of the same type. That does not involve the neighboring different types of cells of different tissues or on the different sites. This type of signaling allows the cells to self-regulate their own behavior or influence nearby cells of the same type, not of the other. So autocrine signaling is important in processes such as cell proliferation, differentiation, and immune responses. And if we talk about the paracrine signaling, in paracrine signaling, cells release signaling molecules, paracrine factors, into the extracellular space, where they diffuse locally to nearby target cells and bind receptors on their cell surface. And paracrine signaling enables cells to communicate with neighboring cells of different types within the same tissue or microenvironment. Microenvironment is a small environment that is present near the extracellular matrix of the cell. Paracrine signaling is involved in the processes such as neurotransmission, tissue repair, and immune responses. In endocrine signaling, specialized cells called endocrine cell release signaling molecules. These are chemical messengers known as hormones into the bloodstream, where they travel throughout the body to distant target cells that possess specific receptors for the hormones. Endocrine signaling allows for the long distance communication between different organs and tissues and plays a central role in regulating numerous physiological processes, including metabolism, growth, reproduction, and stress responses. As you can see in the diagram, in autocrine, and the signaling molecules binds with its own type of cells with own cell. In paracrine, it is binding with different or nearby cells. And in endocrine action, the signaling molecules, the hormones are released in the blood circulation, and then it reach the distant target cell present away from the gland. And if we talk about the primary messengers, are signaling molecules that bind to specific receptors on the cell surface, initiating a signaling cascade that leads to cellular responses. These molecules directly interact with cell surface receptors, trigger intracellular signaling pathways. Examples of primary messengers include hormones. Hormones are chemical messengers produced by endocrine glands and released into the bloodstream to act on target cells located throughout the body. Examples include insulin, adrenaline, epinephrine, cortisol, and thyroid hormones. And if we talk about the neurotransmitters, are signaling molecules released by neurons at synapses to transmit signals to the neighboring neurons, muscles, or glands. Examples include dopamine, serotonin, acetylcholine, and gamma aminobutyric acid (GABA). If we talk about growth factors are signaling proteins that regulate cell proliferation, differentiation, and survival. They play important roles in embryonic development, tissue repair, and immune responses. Examples include epidermal growth factor, EGF, platelet growth factor, PDGF, and vascular endothelial growth factor, VEGF. And if we talk about secondary messengers are intracellular signaling molecules that relay and amplify the signals initiated by primary messengers. They are typically small, diffusible molecules that mediate the effects of primary messengers in the downstream cellular responses or processes. Examples of secondary messengers include cyclic adenosine monophosphate CMP. CMP is a secondary messenger generated from adenosine triphosphate ATP by the enzyme adenylate cyclase in response to activation of G protein coupled receptors GPCRs. CMP regulates various cellular processes including metabolism, gene expression, and ion channel activity. Inositol triphosphate IP3 and diacylglycerol DAG. IP3 and DAG are generated through the cleavage of the phosphodiethyl inositol 4,5-bisphosphate PIP2 by phospholipase CPLC in response to the activation of certain cell surface receptors, such as receptor tyrosine kinases. IP3 triggers the release of calcium ions from intracellular stores, while DAG activates protein kinases PKC, which regulates numerous cellular functions. So, if we talk about the calcium ions, they are also secondary messengers. Calcium ions serve as a versatile secondary messengers involved in a wide range of cellular processes, including muscle contraction, neurotransmitter release, gene expression, and cell proliferation. Changes in intracellular calcium levels are tightly regulated by various channels, pumps, and exchangers. So, some of the extracellular messengers other than secondary primary are amino acids and amino acid derivatives. Examples include glutamate, glycine, acetylcholine, epinephrine, dopamine, and thyroid hormone. These molecules act as neurotransmitters and hormones. And after amino acids and their derivatives, certain gases such as NO and CO, nitric oxide, and carbon monoxide. Steroids, which are derived from cholesterol. Steroid hormones regulate sexual differentiation, pregnancy, carbohydrate metabolism, and excretion of sodium and potassium ions. So, a wide variety of polypeptide proteins. Some of these are present as transmembrane proteins on the surface of an interacting cell. Others are part of or associated with the extracellular matrix. Finally, a large number of proteins are excreted into the extracellular environment, where they are involved in regulating processes such as cell division, differentiation, the immune response, or cell death and cell survival. And now, if we talk about the receptors, extracellular signaling molecules usually but not always recognized by specific receptors that are present on the surface of the responding cell. Receptors bind their signaling molecules with high affinity and translate this interaction at the outer surface of the cell into changes that take place on the inside of the cell. The receptors that have evolved to mediate signals transduction are indicated below. First of all, the G protein coupled receptors, GPCRs. They are a huge family of receptors that contain 7 transmembrane helices. These receptors translate the binding of extracellular signaling molecules into the activation of GTP binding proteins. And if we talk about the steroid hormone receptors, second type, these receptors function as ligand regulatory transcription factors. Steroid hormone diffuses across the plasma membrane and bind to the receptors which are present in the cytoplasm. Hormone binding results in a conformational change that causes a hormone receptor complex to move into the nucleus and bind to elements present in the promoters or enhancers of the hormone responsive genes. This interaction gives rise to an increase or decrease in the rate of gene transcription. And if you talk about GPCRs and their second messengers, GPCRs are so named because they interact with G proteins, members of the GPCR superfamily, and are also referred to as 7 transmembrane helices or receptors because they contain 7 transmembrane helices. Thousands of different GPCRs have been identified in organisms. In fact, GPCRs constitute the single largest superfamily of proteins encoded by animal genomes, including among the natural ligands that bind to GPCRs are a diverse array of hormones. They are present in both plant and animals. Neurotransmitters, opium derivatives, hemoattractants, such as molecules that attract phagocytic cells of the immune system, odorants and tastants, molecules detected by olfactory and gustatory receptors, eliciting the sense of smell and taste, and photons. That is an example of GPCR. These are the 7 transmembrane helices on the left side. It's shown in green colored. Ligand binds with the pocket amino group on the outside, and cytoplasmic side faces carboxyl group. And there are 2 proteins alpha, beta and gamma. Beta and gamma are associated with each other, and they bind with the alpha protein, and that alpha protein is actually GTP binded, whereas the G proteins other than alpha protein are binded with the GDP. And on the right one is the, in purple color shown, is the effector. The membrane-bound machinery for transducing signals by means of salmon transmit receptor and a heterotrimeric G protein. Receptor this type, including those that bind epinephrine and glucagon, contains 7 membrane-spanning helices. When bound to their ligand, the receptor interacts with the trimeric G protein, which activates an effector such as adenylyl cyclase. As the subunits of G protein are linked to the membrane by lipid groups that are embedded in the lipid bilayer. And if we talk about the signal transduction by G protein coupled receptors, G protein coupled receptors normally have the following topology. Their amino acid terminus is present on the outside of the cell. The 7 helices that traverse the membrane are connected by loops of varying length, and the carboxyl terminal is present on the inside of the cell. There are 3 loops present on the outside of the cell together that form the ligand binding pocket, whose structure varies among different GPCRs. Different cells, they are of different type, but they have 7 transmembrane helices, not greater than or less than them. There are also 3 loops present on the cytoplasmic side of the plasma membrane that provide binding sites for intracellular signaling proteins. Means to say, either the stimulus is internal or external, GPCRs are well equipped to receive these signals on the receptors. And if we talk about the mechanism of transduction, signaling transduction through G protein coupled receptors involves a series of steps that occur upon ligand binding to the receptor, leading to the activation downstream signaling pathways. Here is how it typically occurs. With the help of the receptor and ligand interaction, GPCRs are cell surface receptors that span the cell membrane 7 times, forming an alpha helical transmembrane domain. Ligands such as hormones, neurotransmitters, or other signal molecules bind to the extracellular domain of the GPCR, inducing a conformational change in the receptor. That is how all this process occurs. First of all, a hormone binds with the pocket that is present on the outside of the cell with 3 helices. And these growth factors, neurotransmitter hormones, these actually bind on the pocket that can be from the outside, that can be from the inside. And in the diagram, I am going to explain these diagrammatic exclusions in detail in the upcoming slides. The ATP binds and the separation from the alpha subunit of the beta and gamma subunit of the G protein. So that will further downstream GBY signaling, that will phosphorylate alpha subunit, beta and gamma together will phosphorylate the downstream targets. So biological function is what is provided, biological function by these GPCR: cell proliferation, apoptosis, autophagy, ER stress, EMT, metastasis, invasion, angiogenesis, cancer, stemness, drug resistance, tumor microenvironmental alteration. These are all down by these phosphorylations of these GPCRs. So different factors are coming, like in GAIS, GAIO, GAQ11, GA12 and 13. These all will act on the transforming factors that are present on the gene, and the gene expression will be altered. So activation of G protein. The conformational change in GPCR facilitated the activation of G heterotrimeric protein associated with the intercellular side of the G protein. Consists of 3 subunits: alpha, beta and gamma. In the inactive state, the A subunit is bound to GDP carnosine diphosphate, and the beta and gamma subunits are associated with the A subunit. They are attached with the A subunit, as you can see in the previous diagram. They are attached with the A subunit, and in the later on they will be dissociated to activate the A alpha protein or alpha subunit. Ligand binding induces the conformational change in the GPCR, causing the A subunit to exchange GDP for GTP. That will replace GDP with the GTP, guanosine triphosphate, leading to its dissociation from the beta and gamma subunits. The GTP bound alpha subunit of the G protein and the free beta subunit both activate downstream effector proteins such as enzymes or ion channel. The activated effector proteins initiate intracellular signaling cascades by generating second messengers such as cyclic AMP, inositol triphosphate and diacylglycerol, or by directly modulating ion channel activity. And if we talk about the amplification of the signal, the activation of the effector proteins leads to the generation of second messengers which amplify the initial signal. Signaling messengers diffuse within the cell and activate downstream signaling pathways, ultimately eliciting the specific cellular responses. For example, CMP activates protein kinase A, PKA, which phosphorylates the target proteins to regulate gene expression, metabolism, or other cellular functions. And if we talk about the termination of the signal, the signaling cascade is terminated through various mechanisms including the hydroxyl GTP bound to its alpha subunit by the GTPase activity, leading to its reassociation with the beta gamma subunits and inactivation of the G protein. The ligand dissociates from the receptor, allowing the receptor to return to its inactive state and reset the signaling pathway for subsequent activation. So overall, signaling transduction through the GPCRs enables cells to respond to a wide range of extracellular stimuli and modulate various physiological processes, including neurotransmission, hormone secretion, immune responses, and sensory perception. So that was the video for today. We'll meet you in the next video. Till then, goodbye.